A very good morning, everyone. I'm Kapila Shekhawat, and I welcome each one of you to this prestigious 30th Dr. B.P. Pal Memorial Lecture 2023, which is jointly being organized by Genetics Club and Postgraduate School, IRI. I request the dignitaries to kindly take their seats on the dais, please. We are highly honored and privileged with the benign presence of Dr. Himanshu Pathak sir, Honorable Secretary There and Director General ICR as the chairperson of this session. I feel immense pride and happiness to welcome Very Honorable Dr. Swapna Kumar Datta sir, Founder Vice Chancellor of Vishwa Bangla Vishwavidyalaya Bolpur, also former Vice Chancellor of prestigious Vishwa Bharati University Shantiniketan and former Deputy Director General Crop Science ICAR New Delhi as the eminent speaker of today's lecture. I also extend a warm welcome to Dr. A.K. Singh sir, Honorable Director IRI and Vice Chancellor, under whose able guidance we all are marching ahead. Dr. Anupama Singh, Honorable Dean and Joint Director Education IRI, is also welcome to the program. I request Gopal sir to kindly do the honors, please. Dr. Gopalakrishnan sir, Head Division of Genetics and President Genetics Club, is also welcome to the program. Thank you. This memorial lecture, ladies and gentlemen, is being organized jointly by IRI and Genetics Club every year to pay tribute to Dr. B.P. Pal for the pioneer services and role played by him in transforming Indian agriculture. And this is 30th lecture in the series. Born on 26 May 1906, Dr. Benjamin Perry Pal was the first Indian director of this prestigious institute, IRI, who also became the first Director General of ICAR in 1965. Dr. Pal is one of the most distinguished agricultural scientists known for his outstanding contributions in crop improvement, particularly for disease resistance in wheat. Way back in 1954, he developed the wheat variety NP809, which could combat all three types of rust. Despite being a wheat breeder, he is equally known for his special interest in rose varieties. For his remarkable contribution, he has been awarded the prestigious Padma Shri in 1959 and later Padma Bhushan in 1968. The appellation of this very hall as Dr. B.P. Pal Auditorium signifies his contributions and the importance of this lecture as well. We will wish to now begin the program by the invocation song, which is being sung by the PG students of IRI. I request them to come over the stage, please.
विष्णु जिन चरणों को सेवते सूर्य ब्रह्म शिव विष्णु जिन चरणों को सेवते हम शिशु हैं अज्ञानी माता इन चरणों का गार दे वीणा पाणी तार दे तार दे तार दे माँ सरस्वती शारदे माँ सरस्वती शारदे श्वेत कमल तेरा आसन है श्वेत हंस तेरा वाहन है श्वेत कमल तेरा आसन है श्वेत हंस तेरा वाहन है हम पर सिद्ध सृष्टि कर जननी बुध चीरती धार दे वीणा पाणी तार दे तार दे तार दे माँ सरस्वती शारदे माँ सरस्वती शारदे पूर्ण इस अज्ञान उदद से पूर्ण इस अज्ञान उदद से वीणा पाणी तार दे तार दे तार दे माँ सरस्वती शारदे माँ सरस्वती शारदे माँ सरस्वती शारदे माँ सरस्वती शारदे
Thank you all for your kind attention, and now we'll have our lamp lighting ceremony. I request the dignitaries on the dais to kindly join and to start the program, please. Thank you, and I again request you all to kindly pay a floral tribute to the pious soul of late Dr. B.P. Pal as the mark of respectful homage for the selfless service to the nation and to the mankind. Thank you, and with the permission of the Chair, I now request and invite Dr. Anupama Singh, Dean and Joint Director, Education IRI for the welcome address.
Thank you, Dr. Kapila. A very warm good morning to all. On the occasion of this 30th Dr. B.P. Pal Memorial Lecture, on behalf of the Institute, Director and Vice Chancellor ICAR IRI, it's a matter of great privilege and pleasure for me to formally welcome Dr. Swapna Kumar Dattaji, former founder VC, Viswa Bangla Viswavidyalaya, former VC, Vishwabharati University Shantiniketan, West Bengal, and former DDG Crop Science ICR New Delhi, who has been very kind enough to accept the Vice Chancellor IRI's invitation to give the prestigious lecture on the highly relevant and thought-triggering topic, Human Face of Agriculture, Genome Changes with Time. Sir, we are grateful to you for sparing your valuable time. IRI also feels humbly honored in welcoming Dr. Himanshu Pathakji, Secretary DARE and DGICR and Chairperson of this function, to spare his valuable time, as he is always a source of inspiration for all of us. For the UG students of this founder batch, I must, introduce you to Dr. Pathak, that he is one of the most brilliant alumni of IIRI. So, that's a matter of pride for all of us. So we also welcome all the distinguished dignitaries in the front rows. I'll take a minute to name them. Professor R.B. Singhji, Professor Beethal, Professor K.C. Bansal, Dr. R.K. Jain and Dr. Rashmi Agarwal, former Deans of the PG School IRI, Dr. Sadamte, the Honorable Member Academic Council, and many other dignitaries, heads and professors of this institute. It's my duty to briefly mention about this prestigious lecture. So Professor M.S. Swaminathan initiated this prestigious lecture series to commemorate the contributions of late Dr. B.P. Pal. He was of the opinion that the essence of the contributions of Dr. B.P. Pal, primarily in the field of domain of roses, genetics, plant breeding per se, and his persona around IIRI must live in the minds and hearts of everyone in and around IIRI. Dr. Pal is rightly known as father of roses and father of agricultural research system in India. Today's lecture is 30th in this series. Dr. Benjamin Perry-Paul had his earlier education from Rangoon University before he proceeded to Cambridge University, UK for his doctoral degree in the discipline of genetics and plant breeding. He joined IRI Pusa in 1933 and remained associated throughout his life with IRI. Dr. Pal became Institute's director in 1950, remained on this post till 1965, when he was appointed as the first Director General of the reorganized Indian Council of Agricultural Research, and subsequently he superannuated in 1971. Credit for establishing the postgraduate school, which is now rechristened as the Graduate School IRI, goes to Dr. Pal only. He introduced the course credit and the trimester system in IRI's higher education system. This institute is humbled to have dedicated its central beautiful garden to Dr. B.P. Pal on 31st December 1971, as well as christening IRI Auditorium and the Art Gallery after the great soul's worldly name. Dr. Pal's contributions have been honored with many prestigious awards, both at national and international level. Some of them have already been mentioned by Dr. Kapila. Still, he was the recipient of Padma Shri, Padma Bhushan and Padma Vibhushan. He donated major portion of his estate and property to IIRI for the cause of agricultural research in the country. I once again welcome you all on this occasion. Thank you very much.
Thank you very much, ma'am, for that welcome address, and I now request Dr. A.K. Singh sir to kindly introduce the chairperson to the August gathering. Sir.
A very good morning to each one of you. the distinguished speaker of Dr. V.P. Pal Memorial Lecture, Dr. Shrapan Datta, Chair of the function, Dr. Himanshu Pathak, my colleague Dr. Anupma Singh, Dr. Gopal Krishna. In audience, we have very distinguished Professor R.V. Singh, Padma Bhushan, Dr. Virthal, Dr. Bansal, our former Dean Dr. Jain, Dr. Asmi Agrawal, Member Academic Council Dr. Sadamate, Madam Kaur, Director NIPV, and Project Director Water Technology Center, Dr. Brahmanand, all heads of the division. Sir, we have got, with your great efforts, a very new team. Out of 22 heads of the division, 20 have joined. We are waiting for the remaining 2 to join very soon. So thanks for giving this young group of leaders to IRI to take IRI to a newer height. We are obliged to you. This important lecture series, as has been told, is a very prestigious lecture series, and audience we have a large number of students, and particularly our undergraduates who have joined their program. They were admitted about a semester back. They had courses online, but for second semester now they have joined offline. They are in the campus, and this is going to be a kind of lifelong experience for them. So I would like to extend a warm welcome to each one of you for this important function. Dr. Himanshu Pathak is known very well to each one of us. It is his home IRI, and in home one doesn't need introduction. But for the benefit of our young students who are possibly going to hear him for the first time, I would like to tell about Dr. Pathak, so that you know, in Dr. Pathak and also in Dr. Shrapan Datta, we have 2 role models, and unless you have a role model in your life, you cannot aim for the highest thing that you can think of achieving in your life. So these are some of the people that you can always take as role model. Dr. Pathak did his bachelor in agriculture from Banaras Hindu University and masters and PhD in soil science and agricultural chemistry from the Indian Agricultural Research Institute. His research area has been focused on quantification and mitigation of greenhouse gas emission. Such an important topic. You know, Indian rice paddy was declared as a culprit for emitting as much as 37 million tons of methane to the atmosphere. And imagine if that kind of data figure would have continued. Today we are exporting rice worth $10 billion US, $10 billion US. Out of $50 billion agri and allied export commodity, rice share is $10 billion. And if this tag of being culprit of greenhouse gas emitter to the extent of 37 million tons had continued, we would not have been in a position to export. We would have been banned by the international community. But the research carried out by Dr. Pathak, his team, which was heavily led and initiated by Professor S.K. Sinha, the former director of IRI, that this figure was actually demonstrated by wet lab experiment throughout the country, measuring the emission, quantifying it to almost 3.7 million tons, 1/10 reduction, and that's what makes Indian rice, and India is the largest exporter of rice in the world today. So thank you, sir, for that excellent piece of work. And in addition to that, he has a specialization in modeling soil fertility, use of GIS and remote sensing in management of land and water resources, use of isotope and radiations in soil-plant relationship, modeling growth and yields of crops. These are the core areas and domain of his specialization. And he, before taking over as Secretary there and DG ICR, he served as Director of National Institute of Abiotic Stress Management at Baramati, then Director of National Rice Research Institute at Cuttack, the famous Central Rice Research Institute, those days was known as, and of course for very long as scientist, senior scientist, principal scientist and professor in different capacities in the Division of Environmental Sciences at IRI. His international exposure and experience, if you see, he was Humboldt Fellow, he was visiting scientist at International Rice Research Institute and also at CSIRO, Land and Water, Griffith, Australia, also a Boyce-Scott Fellow at University of Essex in UK, and he is one of the most cited researcher in terms of his publication. with a H index of 68, I-10 index of 210, and overall citation more than 20,000. It might have gone up. So thank you. I know having such a distinguished leader as head of the organization. He is fellow of the Indian National Science Academy, National Academy of Sciences India, and National Academy of Agricultural Sciences, and number of awards to his credit. But to mention just two, to begin with, the Young Scientist Award of Indian Science Congress Association, and these awards at the beginning of career are very, very important for one to go ahead in his life and keep motivated throughout. And that's why I'm mentioning the very first award, and of course the Rafi Ahmad Kidavi Award, which he got from the Indian Council of Agricultural Research for his voluminous and quality contribution to Indian agriculture. We are thankful to you, sir, for accepting our invitation to chair this function, and I thank you all for joining large number. Thank you very much.
Thank you very much, sir, for introducing the Honorable Chairperson. I now request and invite Dr. Himanshu Pathak, sir, to kindly introduce the speaker to this worthy gathering, and also request him to kindly deliver the talk. Sir.
Thank you very much, Professor Datta, Dr. A.K. Singh, Dr. Anupama, of course Dr. Gopal Krishnan, Professor R.B. Singh Saab, Dr. Vittal, Dr. Jain, Dr. Sadamate, Madam, PDWTC, Dr. Bansal, all the heads of the divisions, professors, scientists, technical staff, administrative staff, supporting staff, and of course today I can see large number of students, particularly UG students who have very recently joined. As Professor Singh said, it is indeed not a pleasure, it is an honor for me. And whenever I come to this hall, I always go back to that year when I for the first time stood up on this particular dais. And that was in 1988 to present my MSc dissertation thesis work for competition. You know, I like gold medal for MSc and so on. And then of course on several occasions students and so on I came. But then as I said, whenever I come to this particular place, I again go back to my all old days. My congratulations and welcome to all the newly joined head of the divisions. So congratulations to each and every one of you. We are really looking forward, really looking forward for valuable contributions. And also the young students, UG, which have started for the first time. So welcome to this great institute. And not only for the sake of saying it is great, it is indeed a great institute. And whenever you will be here, you will be a part and parcel of this institute, and you yourself will realize why and how it is so great. And today we are celebrating one of probably the greatest among all of us, the scientist Dr. B.P. Pal, by introducing one lecture on him. My job has been only to introduce Professor Shravan Datta. I can go on speaking without this note also, but then let me tell you, Dr. Datta certainly doesn't need any introduction, particularly for this audience, except the students. So I thought I might not be reading all these things, but I then thought I must read it, only to tell our students that all of you and all of us should aspire to have a biodata like his. This is the kind of biodata every one of us should be aspiring for. We may not be able to achieve that, but then our aspiration should be in that direction. Professor Datta is a very well-known biologist, all of you know, with vast experience in plant biotechnology, agricultural science, and global agriculture, including policy and research. He obtained his Bachelor of Science, BSc degree in Botany honors in 1972, just to tell you what is his age, just indicating actually. From Presidency College, of course now it is known as Presidency University in Calcutta, West Bengal. He went on to complete his Master of Science, MSc in Botany from the University of Calcutta in 1974. In 1980, he received his PhD from the same university, and then he went to pursue a course on intellectual property rights, IPR, at Cornell University in the United States in 2003. Several awards and honors, recognitions have been bestowed upon Dr. Datta. He has received prestigious DBT Distinguished Biotechnology Research Professor, Raspberry Ghosh Chair Professor, University of Calcutta in that professor, DAD Fellowship of Germany, visiting faculty at UC Davis, USA, senior scientist at ETH Zurich, Switzerland, global rice crop leader at Erie, Philippines from 1993 to 2005. He is elected fellow of all the national academies, including Indian Academy of Science, Indian National Science Academy, of course our own academy, National Agriculture, National Academy of Agricultural Sciences, and also fellow of Third World Academy of Sciences, and of course there are several others. He is the founder, Vice Chancellor of Vishwa Bangla Vishwavidyalay, Bolpur, Santiniketan, West Bengal. Also, he was Vice Chancellor of Vishwabharati, Santiniketan University. And then, from 2009 to 2015, he was Deputy Director General of Crop Science in Indian Council of Agriculture Research. Besides his research, probably one of the very significant contributions which he has made is guiding the students. He is mentor of many, though officially he has guided 44 PhD students, the number could be a little more, but then otherwise he is mentor of many of us. Let me tell you, for me also he is one of the mentors. And I know Professor Datta when I met him for the first time at Erie, Philippines, maybe around 20 years back or maybe a little longer. And even that time also at Erie, Professor Datta was one of the brightest scientists at Erie. And everybody will know who is Professor Datta, what is his area of research, and how he is contributing. And students will be always looking forward to work with him, not only from India, but from the whole world. His research and development work have resulted in the creation of several biotech products, including golden indica rice, high iron rice, hybrid BT rice, disease, pest and herbicide tolerant varieties of rice, jute and pulses. His contributions to plant biology and agriculture are reflected in his extensive publication record of over more than 150 research papers, appearing almost all the prestigious journals of the world, including Nature, including Science, and so on. Of course, for the record's sake, his H index is 64, ITN index is 153, with citations of more than 1600, 400, and many more actually. I can go on reading and reading, but as I said, he is mentor to many of us, not only those students who are in the field of genetics or plant breeding, but even otherwise also. And besides his scientific credentials and so on, there are few very fine qualities with Professor Datta. I must mention those. He is friendly to all of his students. Age has never been a criteria to him to interact, to become friend. And that's why even 20 years back also, he could take me as one of his students or friends, and till today we maintain that. The second quality is, and probably that comes with anybody who has got some linkage with Santiniketan, is his deep knowledge and interest with the works of Rabindranath Tagore. He in a way is a kind of dictionary of Rabindranath Tagore. All of his songs, writings, novels, thoughts, and so on. And anyone will be really impressed to know the depth of his knowledge in those aspects, besides of course he is master of biotechnology, plant breeding and all of these things. So it is indeed an honor for me, and when I say it is honor, it is really an honor for me to have you as the distinguished speaker of the day, And with this, on behalf of all of us sitting here in this great hall, and the name is of course Dr. B.P. Pal Auditorium, it is an honor for me to invite you to please deliver this particular 30th Dr. B.P. Pal Memorial Lecture. Thank you very much and welcome you, Dr.
Good morning to everyone. Professor Himanshu Pathak, Secretary Director General ICAR, Dr. Ashok K. Singh, Director and Vice Chancellor ICAR IRI, Dr. Anupama Singh, Dean and Joint Director Education ICAR IRI, Dr. S. Gopalakrishnan, Head Genetics, President Genetics Club ICAR IRI. I see lots of my own friends and colleagues, also my seniors, Dr. R.V. Singh, Dr. Vithal, Dr. Bansal, Dr. Jain, Dr. Sadamate, and all the other division heads and teaching communities, people, as well as students, my dear students. And I'm glad that there are many UG students are also present today in this hall. That's a great thing. Actually, after listening Professor Himanshu Pathak, I thought it would be nice now to go home with that kind of kind words that he offered to me. I felt humbled, and I felt now I feel that I shouldn't speak anything. I have no idea that I have done so much or so much things that Dr. Pathak has emotional and passionate touch with me, as well as with A.K. Singh all the time. Whenever I met A.K., I was going through some of my old picture. I found lots of picture I have with A.K. and I'm surprised that in many meetings, and A.K. is a very silent person. A.K. is there in the picture, but he doesn't show much of visibility, although he's a tall guy and one can easily identify A.K. Singh is sitting there. So in many ways that this hall that we are here today on 30th Professor B.P. Pal Memorial Lecture. It's a great hall, as Dr. Pathak mentioned, and I've been here many times while I was working at ICAR, and now I'm here to give I wanted to sometimes to give a talk on B.P. Pal Memorial Hall lectures, and I'm glad and grateful to Dr. A.K. Singh that has given me this opportunity, because this is the man I didn't interact with him much, but I felt something with his charismatic figure and charismatic smiling face. Only one time I met him, but after I listened many of his memorial talk in this hall, so I've been listening of Professor B.P. Pal. It tells me that I'm very close to him after listening many times with such a great personality of ICAR. The first slide shows that a few things together is kind of keywords probably. And I move on to my next slide. As I mentioned that if you can recognize the picture, that is Dr. B.P. Pal. This is Archana Sharma. Professor Archana Sharma introduced me to Dr. B.P. Pal in 1978 at Ahmedabad Indian Science Congress. And some of you may know this is Dr. Kauravi Datta. So that was my one time that I met Professor B.P. Pal in 1978. Many of you probably was not born even when I just had the opportunity to see him. This is the same hall. In front of this hall, some of the eminent Indian agriculture scientists are here. I thought I should, because it is Dr. B.P. Pal Sabha Bhavan, I thought I should include this as incidentally some of the people, as Dr. R.V. Singh is here. It's great that this picture I have with me. Well, then I would take today a journey of agriculture from time immemorial with sort of philosophy of humans and as well as the agriculture perspective. And I would link with genome with this agriculture growth and see how with time everything is changing. And also, before coming here, I had a brief discussion with Dr. A.K. Singh. Even some alarming things are also now coming up for future with artificial intelligence. Anyway, let's see how we move on. So 2,000 to 30,000 years back, how the different crops have been evolved and they are being domesticated. The oldest one is from Italy as well as from Israel, and it's oat. Now, how we see the genome is a complete set of genes and genetic material that present in cell and organism. Now, today, we know that all the evidence show that early RNA protogenome that gave rise to the DNA genome of human beings as well as all the plants and animal organisms. And this is single origin. So this is now, you can now go back to the ancient time and see how the things have changed and moved for the future. Now, another interesting component that has come, that we know that evolution, adaptation and speciation is a continuous process. But interesting part is the genetic code is also evolutionary expanding. The particular work on Euplotes crassus that shows one EGA codon that could encode 2 amino acids, cysteine and selenocysteine. It could read in both ways. That means the genetic code can be evolutionary expanded, which is a good thing actually. It's not that that environment is changing, this is going in a bad way. Our genome is also expanding, genetic code is expanding. So there is a parallel movement is going on so that we also need to be evolved or going to evolve. That's a good thing, nothing wrong in it. And then there comes the genetic or genome editing. Now what is that? It's a simple the molecular scalpel, the CRISPR-Cas9, and you cut on and figure out and put it again and rebuild it. It allows the rapid mutagenesis, and this mutagenesis would help you in a precision plant breeding. So that means genome editing today would go on rapidly in a very expanding manner and make much more new traits evolved, and modification of the traits would be involved or evolved, and eventually it's a good thing for the plant breeders to move on. Earlier, the Green Revolution, we know that. We know that it is SD1 or RST for rice and wheat, respectively, and that the functional of this gene reduce the apical dominance of the plant, resulting in multiple tillers and giving high yield. And today, this green evolution, what is happening, making India surplus or exporting, as you heard from Dr. A.K. Singh, in rice itself we're exporting $10 billion US, and overall agriculture produce exported 50 plus billion US dollars. 50s earlier we are in a begging bowl and importing and asking for support, and today we are in exporting country. The good thing that at that time it's not Norman Borlaug who developed this entire things of Green Revolution, success of Green Revolution, and it happened particularly in India, and C.V. Subramaniam is the agriculture minister, and that made everything happen in a befitting manner. Green Revolution success and the latter part, the last 1 or 2 decades, many countries particularly started talking about zero hunger, and particularly Brazil even explained it and make a statement of zero hunger. But I'm glad that it is not zero budgeting, and that might siphoning all your money and not giving much. But Zero hunger is a good thing, and we should achieve it. The problem is that Indian government made a lot of efforts to give rise to very sort of cheap rice, wheat, and etc., etc. But our hunger index doesn't tell the same story. It just tells an opposite story. If you looked at in 2000, a few years back, it was even 103 out of 119 the hunger index, and today even it is 107 out of 100, maybe 21. That means something went wrong that we need to figure out what went wrong. Tagore, as Dr. Pathak mentioned, that I have kept particularly a few slides, that you know that Tagore is a poet, he is a philosopher, he is a music composer, he is an artist, and he is a, he wrote so many different kinds of stories, and his writings on nationalism needs attention. But I'm not trying to say anything about that, his literature. And today put a few slides, Tagore as agriculture visionary. He realized, he traveled extensively in Bengal, Bangladesh, and some of the neighboring states, as well as he traveled even 30 plus countries. Even at that time, all the travels used to be by some kind of ships. And Tagore saw only the villagers, or particularly the farmers, are extremely poor, and they live on agriculture for their living. And then they do not have any idea about the modern science or any technological know-how, how to make agriculture a better way. So Tagore didn't like it. Tagore thought that majority of the people are poor, and then they have nothing to do, and we need to change it. So he started plowing festival, so that where all the people could come as a first, as a kind of entertainment, a bit of small business, and then the festival need to be attended. Long back after post-independence, I think Atal Bihari Vajpayee also attended one of these plowing festival in Shantiniketan. There are many eminent people went there for this particular festival. It's still going on, and it is still there. The second part that Tagore started is cooperative agriculture. He realized that all farmers independently, they can't do much. So they have to work together. Even 100, 120 years back, he started cooperative agriculture with the Grameen Bank of his own money from the Nobel Prize, as well as other money that he has, his Jamindari money. He put the zero interest money giving to the farmers, and so that they can work on it. Third component that he initiated, I don't know whether you can recognize this young Prashanta Mahalanobis, the father of Indian Statistical Institution. When Prashanta Mahalanobis wrote an article on the water flooding, etc., there is a pattern. So Tagore read that article. He invited Prashanta Mahalanobis to come Shantiniketan, and Prashanta Mahalanobis worked with Tagore for 3 years. The task was how to predict that water flooding and the excess or rain and those kind of water, how to store the water, excess water, and how to use that water for irrigation and for other purposes. Even at that time, say 100 years back, Tagore looked into that aspect. Unfortunately, lessons still not yet learned properly. And still we have a flood, we have excess of water, and there are places where we don't have the water. And then the fourth one is the model village. Integrated farming, these are the things that are very much well known. Tagore, I just quote unquote from Tagore's own writing here, because that would make very clear. If we could free even one village from the shackles of helplessness and ignorance, an ideal for the whole of India would be established. Let a few villages be rebuilt in this way, and I shall say, they are my India. This is the way to discover the true India, Rabindranath Tagore. Still we are hearing about model village. We have smart village as well as all the technology, know-hows are there. But still Tagore's dream of one model village, we still need to wait when we see that one. And that sort of thing is still missing, and we have to get it. The fifth one, at that time, Tagore realized that the farmers do agriculture with fragmented land, small pieces of land. And he said, even it is written in Bengali, who could read Bengali can read it. He says that even bringing water in a kind of, with the holes, and you put water in the land. That's nothing to do. So he looked for the consolidation of land even at that time. Even today, if you say that, people will be jumping on you. Oh no, don't talk about this, because we believe in smallholding farms. We believe in this. We have to keep that way. Yes, I believe that India is a country of diversity. India is a country of different kind of resources. We have to keep smallholding farms and to be there to a large extent. I agree. But there are places, Punjab, Haryana is giving the good example, where consolidation of land is happening, mechanization is happening, everything is happening in a different way. The water, drip irrigation, etc., etc. So with a combined harvester and a huge land of when huge land is available and to grow in a massive mechanized way, the growth and productivity would be much higher. Now, as an example of, you may have heard the Shingur in Bengal. It's a huge controversy. Tata, Mamata Banerjee, etc., state government as well as opposition, etc. Now, how many of you would know that we are following still 1894 Land Acquisition Act, and that gives 997 out of 1,000 acres of land given to Tata to build up the Tata Nano Company factory. 15,000 people live on that 1,000 acres land. 15,000. Nano is gone, fertility is gone, everything is gone. That Shingur, still Shingur name is there, nothing is there. The model is, say these are the fragmented lands, you could easily carve it to make it a composite, still a few more units, different crops can be grown, industry can be incorporated there, and that could make a tremendous, interesting and functional model. This is a challenge. Well, now I would I will move to a different phase now. 2050, 10 billion population of the world. India is already now number one in the population, and we will be 1.6 billion during that time. So this is the one I was mentioning about the cost of this Food Bill Act, 2.38 lakh crore per year. And many state government even reduced that price also, even not even 2 rupees rise, 1 rupee rise, even 0 rupee rise given to the farmers. Everything is fine except this one. India hunger index still remains 107 out of 121 of the world. Where is going then? Government policy is fine. Roadmap is there. Implementation, there is something a serious issue that need address. Otherwise, everything what you talk, what you work has no meaning. Chetan Bhagat, I think young people definitely know as a best storyteller and story writer. Chetan Bhagat 2013 wrote an article where he said that if we go on with this kind of policy and do not see that it's functioning well, then we would be going for, people will be more poorer than where we started. It doesn't help in that way. Swaminathan, 1988, the famous book Science, Ethics and the Food, after receiving his World Food Prize, then there was a conference, and that conference Swaminathan was there. Many people, Amartya Sen and many other different kinds of people were there together. There's a small book. If you read it, you will find a lot of interesting articles in there. And in that book, he mentioned that the improved technology is definitely help farmers, but it would not work until the public policy is in place in a proper way. So there are two things: science, technology, public policy, implementation, and there is a gap. And all of these things you need to keep it in mind that it's not only science alone, but there are all of the other things need to put together. And 20 years back, when Atal Bihari Vajpayee, the Prime Minister, 88 Indian science foundations said 1/3 of the country's children were malnourished. And today, children plus. Not only 1/3, 1/3 plus young children and women are still malnourished. So when you say something, or when our government also knows something, and we also believe that, but there is a still gap that we are not implementing certain things in a right manner to help it. Now, I put now a kind of summary that the next minutes I could go on that. Challenges: population, limited natural resources, more foods required. These are three main things that we have in mind. And possible consideration that everybody know that we have to increase the productivity, water and fertilizer use efficiency, reduce post-harvest yield loss and market use efficiency, real-time agriculture growth, digital agriculture, right policy and management for remunerative agriculture, science and technology including artificial intelligence, particularly genomic tools for agriculture growth and sustainability. The last part is to all of you, that ICAR, SAU and all Indian institutions, they have done tremendous work. We are grateful to ICAR, we are grateful to all of the people that who did so much of work. They must continue that good work. They must continue that work irrespective of any other pressure from anywhere. Because that is very important. This is for us, this is for you, this is for the next generation. They should not compromise the science and technology for anything else. And the other challenge is that our ecological debt is increasing, continuously increasing. Because simple, 6% global ecological footprint, 4% global biocapacity, and 17% of the global population live in India. So that's a challenge. This is definitely challenges. We have to do everything in a different way. And these challenges we have to accept and move forward with more efficient way to work. Again, I'm bringing back this slide because that now after domestication, now the genomic tools allowed us to know that the different genes and QTLs are associated with the different traits. And these are the important components that are associated with all our wild species as well as the plant that has been domesticated. 1865, Gregor Mendel. I think Dr. Arveshi would be very happy to see this kind of Mendel's picture and with the genetics concept. And then this is moving on. And one thing we must salute to our ICAR and IRI, so much significant work has been done on Basmati. And Basmati made a tremendous success and joy for all Indians, as well as we should be proud for that particular work. And continuously improvement is going on. Now the success story is that land races 1900 and it's keeping on. If you read these things, and it's moving on, and the potential yield is now 14 to 15 ton per hectare. And it cannot be more than that because harvest index does not support anymore. So that we have to limit, mathematically we have to limit certain aspect of the yield, but then other management need to be put together so that we can reduce the post-harvest yield loss. In case of wheat also 1850 and continuously there is improvement going on. All different kinds of agriculture practice are involved in it, and eventually biotechnology that allowed today different disease and pest resistance, herbicide tolerance, etc., all are integrated in wheat improvement program. Indian wheat scenario. It's lovely to see this slide. It's continuously increasing, and even Rajaram in few years back when just before he got the World Food Prize, he said that it will be difficult for India to achieve 100 ton per hectare. It is because he doesn't see, but I'd be happy to see Rajaram today with us, and today he has tremendous contribution, as well as all Indian wheat scientists did so much well that we are now beyond our target, and it is continuously moving further. Maize, the same scenario, Teosinte to the modern maize, in continuously improving and improving, and this is the good thing that what you see. A few people here I just put it. The World Food Prize winners, Henry Bichel and Gurdeep Singh Khush, for the young people to look at. These are the people made tremendous contribution in rice improvement. Young Langping and Monty Jones, I'm sure some Indian scientists from India also been also World Food Prize winners. Sanjay Rajaram in wheat, as well as Dr. S.K. Vassal for maize. Also, Dr. Lal got also World Food Prize. I think this is even often we see that recognition doesn't come very well or not so easily for Indian. But the real work, hard work, and in many cases, many Indian scientists definitely awarded, and we have to hope that good work always get recognized and get awarded. Conservation of the germplasm, either at ICRISAT or MDPGR director is sitting here, and everywhere in the world, I think it is very important component because these seeds eventually would help us for moving. And this millets 2023 is the International Year of Millets. And when I was at ICR, I made sure that our institute, Sorghum Institution, should have roti that I can, and I made them. I ate sorghum and millets roti at institution, and today I'm sure it's much more progress has been made, and this is our own rich, nutrition rich agriculture crops. And we must do a lot for the millets to be more popular and to be utilized by the people. And also it is also linked with the lots of tribal population and the village people, they live on millets. And it's and from the environmental point of view, I think Dr. Patel would agree with me that this is the crop, it's a very natural to climate resilient crop and to go on further. Same with the germplasm evaluation. Bansal did a lot of excellent work on chickpea and as well as wheat germplasm evaluation, and there are many of those material are still in the gene bank. If we do not use the material from the gene bank, if we just keep it with the gene bank, 50 years from now, that would be no value of that material. And it is better that we should evaluate and use all the material available in the gene bank. And once I made that statement, and some activists took it in a very different direction. I said everything would be going out of the people. And they wrote to Swaminathan also. The Swaminathan said, I don't believe that Chopin can say this kind of thing for the people to take out our germplasm. But he wrote me back and he sent me the letter that some people, how misinterpret the utilization of germplasm in a different way. Nevertheless, this erect type of chickpea is very important for future design of this crop. As we are here today with Dr. B.P. Pal Memorial sort of lecture, as well as to remember him, his work, everybody know that many of the work he has done, but many people know that his love for rose and the colorful rose. And today what we see that it's not only rose, any flowering plant or the color of the plant is based on certain pigment biosynthesis. You can change one pathway, one component from here to here, here to here, here to here, or changes. You change the color. This color does, without so much of light, you don't see much well, but it's a carnation. Blue carnation is a transgenic blue carnation. I have seen that flower myself in US, and blue rose. And today, any kind of color you can have it and you can develop it. And it's not very big science. It's very simple science. change of one enzymatic pathway that could make. Now the Ruby, one of the another reporter gene now makes the colorful pigmentation in the colors in the plant itself, and you can also have today maize and other plants. And what I'm interested to see whether jute can got this color and cotton can got the natural color, so that we don't need to dye. And I know that Shubhabrato Majumdar, who did PhD with me and now he's working as a DVT scientist, he's working on it, and he is hearing that he would be tempted to do some more work on jute. And I think these are the new things that could be interesting to work on it. Jute has another component that it consumes 15-ton carbon dioxide and releases 11-ton oxygen. That is, it is a higher carbon credit efficient crop. That's a very important component. If we keep these jute and allied fibers growing a massive way with more other directions, that would help for the environment. The cotton, good and bad both, 95% are BT cotton. Now any field, anywhere you go there is simply BT cotton. One GM crop in Indian field is moving very nicely without any problem. But the problem is that 2016, 17 and today, there is not much changes of the yield. It is still better than the non-BT, but did not go further and further. And so now the new invention or the new discovery of the genes or new traits or the new incorporation of the genes not being taking place in cotton. Now, who to be blamed? The scientists not working, or our policy not allowing to work, or etc. etc. I'm not blaming anybody, but I'm just raising the issue that India also export cotton. We need to move or to do better crop cotton and to make it better, so that we need to incorporate a new dimension, technology, and the genes incorporated in cotton. Anything we see, there is a sequence, and that what genome sequencing today from manual slab gel. When Banshal was young student or PhD student at the time, definitely he used to do a lot of slab gel work, and many of us did that. And then it goes on moving, third generation sequencing. Today we have single molecular real-time sequencing, Oxford nanopore sequencing, and most cost effective, very simple and straightforward. That means a tremendous progress has made on this area. Now look at one crop rice, 3,000 reference genome sequencing is going on, international level. Then looking for the promoter and different genes, and then genome editing, making even further elaboration of trait improvement. Sequencing, characterization, editing. All things is moving simultaneously. Also, large number of different genome has been sequenced, the pigeon pea, chickpea, palm millet, you name any, groundnut, you name anything. Most of the things have been done, Rajiv Bharsane and his team, and worldwide many people have done all this work is moving on this direction. In jute also, there are 3 publications, including one from Kryzat, Devabrata Sharkar and his team in Kashin. So they also have done draft genome of Corpora solitorius, etc. Now, interesting part is this genome sequencing is now done, and then there is a possibility of a tremendous incorporation of improvement of the traits can take place. Because jute, as such, the crop has not gone much improvement because compatibility is not there. That's one problem. The next generation of genetics in plant, we know the sequencing work, we now know the characterization, but the phenotyping is still a challenge. Why it is a challenge? We need to work hard. And phenotyping is not that easy. Artificial intelligence will not do the phenotyping. You have to do the work and then get the data. Once you get the data in your cloud or in data bank, then maybe you can ask somebody to do the work for you. That's a huge challenging work. I think for the young generation of the students, one need to understand that the genotyping today has become more easier than the phenotyping, but if you do not make phenotyping, then your genotyping would not help much. You know this young gentleman, and then not only that, I just put this slide because I wanted to talk about something on DRS herbicide. DRS, it is not, Bashmati, everybody know that his contribution, but the DRS herbicide is tremendously an important component, and also IRI as well as Savannah. So I think there's a new sort of development is coming up, that direct seeded rice with herbicide tolerance. So this could make a big changes in Indian, at least in rice scenario. And these are the stories, are very well evidenced. IRI people all know that, it has been field evaluated here. So definitely that mutant is extremely doing well, and so we need to see how farmers adapt this material as quick as possible. Now the thing is that HD3406 is a mass version of HD2967 with a different kind of resistant genes have been now incorporated there. So now the science application is getting a very sort of routine straightforward, which is a very good thing, and it would add more value to the trait development. In case of maize also, nutrition maize with the provitamin A, tryptophan in protein, lysine in protein, also CRI, that is NRI, Dr. Pathak and Dr. Mahapatro, when they also developed a good protein-rich rice. That also is a new development that has come in recent time. Now I'll talk a few 2-3 minutes on built-in plant protection. I have not included anything on shade blight and disease resistance. We have done a lot of work, but I did not. Most people are familiar. I have included some slides which I love it, because this is very fascinating to me. One is in the field with grown the BT hybrid BT rice, Minghui 63 in in China. This is the picture. This is the joy that I was telling today in the morning to Ekeshing. When Ekeshing goes to the field, or any breeders go to the field, see his material, just love it. And so this was when I saw our BT rice in China. This side is a control. You see all the pests, leafholder that attacked entire field, and in this side is the BT Minghui 63. No disease, no pest, nothing. So I thought it's fantastic. One thing, there are many reservations for China that we may have, but one thing in China is quite clear to me, that the laboratory science in Europe and America is fantastic, lots of freedom. In China, they put a lot of money for the laboratory research, but they have a lots of freedom also do work and field evaluation in the field. They don't have much restriction. If I want to send some material to China, it takes only 1 or 2 days. They clear it and put a red stamp. Red stamp means not no. Red stamp means send the material. And they start working immediately. So that's the freedom that I was bicycling, seeing the field, and also the work. So that sort of things, I think that may help them in progressing. This is in India, the bacterial blight evaluation in Philippines, and etc., etc. So XA21 and BT rice, this is a very beginning, say 20-25 years back, this work moved to the field and worked very well. Now many of us, many of you, probably young people may not know, but should know that this is Ingopotrichus peter Bayer. The golden rice was developed as a provitamin A rice, and then we made it double haploid because I was very fascinated with the haploid induction in haploid work in rice, wheat, maize, and barley, and particularly in rice it worked very well. We developed homozygous double haploid with the golden rice, but still. These things are not moving as the way we should have loved. But fortunately, Philippines is already made a field testing clear. Many countries now gave the green signal. They can import golden rice, they can consume, or they can also market it. And in Bangladesh, also in the final stage, they have field evaluated, etc., but it's the final stage for releasing the material. So at least is some movement is going on. This is the one side, one sort of commentary that published in Current Science. Many of the people are involved here, including Dr. R.B. Singh, Dr. Dhillon, Dr. Gautam, Dr. Karihalu, Dr. Mahadevappa, C.D. Mai, Padmanabhan, Parida, Dr. Paroda, Manju Sharma, Dr. T.R. Sharma, N.K. Singh, and then Shonti, Tyagi, Varma, Beluthambi, etc., etc. I think indecision of the government policy is very much delaying the biotech on science component. So I think that was the urge that very well written this Current Science review, and so that one could read it that what are the issues and what are the concerns that we have and we should address. The last part is genome editing. I hope I'm still keeping my time. Genome editing definitely is the most exciting tools today that we have in our time. But no matter what are the new things that are coming, they're basically plant breeding. Basically, simply plant breeding. It's the new names, new ways of handling it. You see the plant breeding, conventional breeding, you combine, you cross, and you select. It takes a bit of more time. Genetic engineering, you just choose a particular gene for a disease register or whatever it is, and incorporate it, select it, and takes less time. In genome editing, there are many ways. In this case, it is shown that you have a susceptible gene, you disrupt that susceptible gene, and then it becomes a resistant. So in one straight way that you can make a genome edited materials available to us. So this is again, to me, is the plant breeding. It's a new name. Even the mutation breeding, someone said mutation breeding is different. No, these are all plant breeding. Ultimately, you have to select it in the field. Ultimately, you have to select the material, process the material, goes to the consumers, etc., etc. Herbicide tolerance would be big weight would be coming with the genome editing in all kinds of plants. Kutub Mullah wrote this article. Rapeseed, corn, rice, wheat, soybean, etc. In them any plants, because the gene is there. You just need to incorporate it, make it functional, and or reshuffling it. And that's the way that you can make a genome edited herbicide tolerance plants. And of course, Dr. Pathak would be happy that to see the mitigating the climate change, particularly methane. It is possible now the barley Shushiba gene that would definitely shift the carbon flux favoring allocation of photosynthesis to the above ground biomass over allocation of roots. That way reduce the methane gas emission. Wonderful thing. And what else you need, in particular in case of rice. And there are products now, less browning mushrooms and high yielding drought tolerant maize have come with the genome edited, and these are the potential, all the crops, eventually all major crops and horticulture crops now would be available with the genome editing. So this is a direction that we see very clearly. Artificial intelligence. Is it something new that we didn't know it? Actually no. Everything we knew. What is this? The crop, sensor, platform, data, decision, everything is known. What is the difference is that all the data are being combined, consolidated. Decision making, some software is in charge, taking the right decision or taking certain decision, what you are expecting, what you want. And actually actuation. Actuation for something to happen, they are taking the decision that how to data manage, how you can manage the data. These are the entire things of artificial intelligence. Now in this case, the horticulture crop is very much would be required immediately. So one is the digital world that where you know all the information, what the material is available, material to be moved, material to be shifted, and then one the physical world. The container, the carbon dioxide level, the temperature level, etc., all different kinds of things are particularly to keep the increase the shelf life for the horticulture, perishable fruits or vegetables, so that it would itself will take a decision and adjust the environment of that container, so that some material move for 3 to 5 days and they would adjust accordingly and then take your material, reach to the destination without perishing, without any damage. So artificial intelligence would help us taking efficient and smart decision, or would itself will monitor the entire things and will adjust accordingly as it is needed. So it's nothing but very special, but at the same time it's an efficient way to manage all these things. Similarly with the rain gun. So that we know the drip irrigation is going on for a long time, but the drip irrigation has one limitation, that it is already, you have placed it, it doesn't move. But with the rain gun, it's flexible. You can move the entire things to one place to another place, and with the time bound, and whenever it is required, the water will flow from one place to a very long distance. So that you water it for a certain period of time, and then you stop it. So that again a smart decision would be taken by artificial intelligence, how to use the water in an efficient manner. So this entire thing is the model. That is the cloud storage and processing, that is satellite-based weather data, then real-time decision support, agriculture, robotics, etc. All of these things, eventually all the data that is important, the data would be processed, and then a smart decision to be taken, and that smart decision ultimately, eventually would be acting on actuation. And that's the way the entire things will work. Climate change, definitely, Bansal is the co-author of this review, and climate change was there, is there, would be there. And we have to monitor it continuously and to take a smart decision that how to address that this kind of climate change and what we should be doing. The future crops like C4 rice is water efficient and temperature efficient and nitrogen use efficient, and could be a certain way to move on. There are many, I just put one slide of C4, there are many biological nitrogen fixation, etc., all are the possibility. Now, if genome itself, what we talk about genetic engineering, genome editing, etc., that would give everything? No. Warren Buffett and the Bill Gates, two rich men, they realized that many evolution is successful and gave very good food production, but soil health deteriorated a lot. So the healthy soil is needed. So they put a program on, what is this? Brown Revolution? Brown Revolution program. So they're spending billions of dollars, not talking much, but this experimentation going on. So that how to enrich the soil fertility in a better way, so that with the genome editing and all other aspects, the soil can also help. This is one of my last slide, a few slides I have. The water, I think this is, if you read this one, you would love it. The humans move from, our ancestors move from one place to another place. The plants move, the animals move, all the migration took place because of the water availability and water utilization. Today Israel, almost 85% of their water they recycle it. California now 20% of their water is recycling. Every country is now realizing water is so important. Basically water is the driving force of all nature. A more sustainable future can only be achieved by concerted actions on water starting now. So if you read this review, you would love it. It is very lucid language. It is written. What I'm saying, it's a very important component. We have to use water efficiently and recycle it, so that our with the genome, with the fertile soil as well as water can go together. Now, this interesting article that it says that advancement of photoredox application towards artificial photosynthesis, water splitting and photofixation of carbon dioxide would give you clean environment. There's lots of work going on that aspect. So I think we should not be worried about this and that. There is always science is going on. One good thing about science is global and neutral. Even if we are shortfall with certain things, we can understand that global science and can utilize it for our own needs. So that's the exciting part of science, and we should encourage to know and to use that sort of science for our better life. Well, when all of these things have lots of business issues involved, ethics definitely would be an important component. Particularly universities now having agreement with the different kinds of companies, IRI, ICR, having a lot of now this kind of activities going on, which is very encouraging, very positive. Nowadays we want to open up, we want to have a more understanding and working together. And then what happened actually, that's the interesting part, that Carpenter and Doudna got the Nobel Prize for their genome editing work. These two ladies from different places, they got the Nobel Prize for their genome editing work. History is started long back, but most of the patents owned by Feng Zhang, and Feng Zhang and his group and other groups. Now what will happen now? There are hundreds of patents on genome editing. So as such now we are excited about this work and we'll be all working on it. Now once the product would start coming and commercialized, then we have to see how the story goes on, how we can make best use of this technology for the benefit of our agriculture produce. But it is still an issue of conflict of interest would be there. Now here is Joseph Stiglitz, Norman Borlaug and Muhammad Yunus. Three different people, three different kind of concepts, three different kind of philosophy. Norman Borlaug, we all know that, fantastic, worked and gave it to everybody and freely moving things, and that's the way Green Revolution was so successful. Muhammad Yunus, definitely communication, cell phone, etc., that made women's empowerment, that helped women and particularly village people to understand and to make a better life, which is also very important and interesting. The Joseph Stiglitz, I had to contradict with him. I did not agree with him what he said to me while he was visiting Erie. And then he said the CGIR's concept is, I believe, my dream was CGIR's concept is to develop the technology, use the technology know-how, product development and making awareness to the national countries as well as giving away their material to them. Joseph, putting hands on my shoulder, he said, I don't think what you said that it is true. I said, why it should not be true? All the mission, vision, it is written that it has to be like that. Then he explained to me different IP and different other components and conflict of interest and material agreement, etc., and economics, different kinds of economics behind the story. And if I next time meet Joseph Stiglitz, definitely I will tell him, I'm sorry that I did not disagree with you. I have to agree with you. The things, the way we wish to have, it doesn't work like that. But one thing definitely does work. Your scientific contribution, your publication, and your own work, what you believe. That is no way that can take away or no one can take away. But there are business issues definitely does not allow you certain things to move on which need to be taken in a different way. And Joseph Stiglitz, initially he wrote the book, Discontent of Globalization. He's of that mentality that he always liked to support people. And people, particularly in agriculture, they should get the full support. But then there was a lot of criticism, and later on that he wrote this book, Making Globalization Work. He made some kind of how to make globalization. Globalization is inevitable. Everybody know that. It is there. So from this content to make it work, and he made this kind of stories. Again, back to Tagore. And if I say Tagore was one of the person, also had some connection with the genre of editing, you'd be surprised. But I made it. Either you believe it or not, that doesn't matter. Tagore, in the initial stage, used to write, and every time he write and he doodles. He keep on doodling. He keep on doodling. And those Bengali who can read it, it's a nice story written there. How you recognize a person, whether he fails to understand a person right or wrong, or if he understands a person correctly. He wrote it very philosophically, very nice thing, but that would take a long time to explain it. But what I found, these doodles ultimately convert to beautiful paintings. So paintings, ultimately, that's what we like and what we love. So the genetics, plant breeding, and all kinds of combination, recombination, etc., reshuffling, correction, deleting, everything you do it by genome editing, ultimately to give a good shape of a nice product. So ultimately, that's the product that a beautiful painting that coming from doodles, and ultimately doodling to genome editing. The last slide. Well, in my talk, what I wanted to say, that human face in every perspective of civilization is linked with agriculture, food and feed and etc., and migration, adaptation with water availability and cultivation. It's all connected. You go to their ancestral time, and the way people have dresses, the way people have food, etc., and then mind and etc., everything connected with the food. Even Aristotle once said that food is the medicine. So this entire thing is connected. Well, but for us, definitely we should look that let's care and improve genome, the key architecture of agriculture. Agri growth in India is enormous. How to reduce the gap of potential and actuation. That's the biggest challenge to me. While I was preparing this slide and everything, I'm looking and reading and I found that everything we did right, but then where is something wrong? That means something, there is a gap, that gap we need to find out. I was listening to Dr. Himanshu Pathak on other day, Foundation Day. 2047, that's a big slogan for us, that we should have this target. Why should you wait for 2047? Gandhiji said, do it today. Your future depends on today, what you do today. So then 2047 is too long from now. Today's time is very fast moving, and we must act something today so that we can achieve much more. We are only, AK was telling that $50 billion we are exporting, and that's not even 2 to 3% of total export of India. Netherlands, a small country, export 15-20%. Small countries are exporting so much of billions of dollars. So India's potential is tremendous. We are not realizing, we are not capitalizing it. So why we are not exporting or become a big exporting country, and then we can take the leadership role, even helping other countries with the food for the people. So I think it's possible. Okay, congratulations IRI for NRF #1 ranking. And also I wish worldwide, in true sense, IRI should be the global leader of agriculture. Once again, thank you all for giving me this opportunity to talk to you. Thank you very much.
Thank you so very much, sir, for your words of wisdom. You highlighted the importance of gene editing, and so well you linked it with our goals of achieving sustainable food systems and zero hunger. Describe the journey of agriculture from Tagore to today, from land consolidation to possible considerations, challenges of ecological depths and changes desired, and also showed us the path as to how to move from green revolution to gene revolution. You also ignited our minds that when we can have blue rose and blue carnation, why can't we have golden jute and golden cotton? And finally, everything rests upon the best natural resource management, be it soil or be it water. So ladies and gentlemen, I am sure you all will agree with me that we can have no one better than Professor Datta sir for the cause, and that's why I enlist each one of you in taking a moment and put your hands together to give that amazing speaker a round of applause. Thank you very much, and I now request the chairman of the session, Honorable Himanshu Pathak sir, for his expert remarks. So please.
Thank you very much. Of course, this is a memorial lecture in the memory of a great man, and as I said, probably the greatest among all of us. And today's lecture was delivered in the equally great way by another great person, Dr. Swapan Kumar Datta. He not only talked about science, and while sitting there I could really realize that a scientist has to be a philosopher, and that's why the Doctor of Philosophy is there. Of course he has to be a scientist, or she has to be a scientist. He also has to be a realist. At times he has to be a poet also, maybe art lover. And certainly he or she has to be an enthusiast with lots of hopes for the future. And today's lecture, my colleagues, all of you will agree that it had all of these ingredients. He started with all this history, went to villages, brought in Rabindranath Tagore, then led us to the journey of science, starting from Mendel till today and what we should be doing in times to come. So, sir, thank you very much for such an illuminating presentation. And delivering in such a great way. And of course, this is very much expected from you. But let me share with you few things about Indian agriculture. Indian agriculture is really complex. India itself is really complex. It is not so easy to understand. And to solve India's problems, particularly problem of Indian agriculture, is very difficult, very complex, very challenging. But in spite of those challenges, if I have to divide the entire period after independence till today and then next 25 years, you can divide the whole period probably into 4 parts, 25 years each. So the initial 25 years after India's independence, we spent on what they call as putting the house in order. We had several challenges. So we spent around 25%, the first quarter of after independence, in putting the house in order. And then the second quarter, we moved for increasing food production, sufficiency, and we brought in green revolution, white revolution, blue, and so on. The next 25 years, up to 75 years, and just last year we celebrated that, we could really consolidate of whatever we could achieve in the second quarter after independence. This year we had 330 million tons of food production, around 350 million tons of horticulture production, we are number 1 in milk production, and several others. All of these are the results of those second quarter of our after independence, which we started, which are now bearing fruits. Now the next 25 years, and this is the period of Amrit Kal, what we say. It is not that we shall start working in 2047. The work has started. Work has started for the last few years already. And these 25 years are going to be the period when this country really has to take another very high jump. Very, very high jump. And that is possible. And one hope or the ray is the science. As you said very rightly, science is neutral, science is global, and science is only the hope by which any country, be it developing, underdeveloped or developed, can really lead it. And how best we will be able to utilize science for the benefit of Indian agriculture will be a real challenge. What kind of policy we bring in, what kind of development we bring in, and that will be deciding what we are going to achieve and what this country is going to be in 2047, plus minus this way or that way. And for that, research is important. As I say that today's research is tomorrow's science, is day after tomorrow's technology, and then the next day it will show its impact. So it is not a matter of 1 day or 1 year. You have to start, and we have started fortunately. We are already in this journey. So only the good things are going to come. Several options Dr. Datta showed, including genome editing, digital agriculture, artificial intelligence, and n number of things are now there. Of course, there are challenges. But probably this country has already taken a decision that we are going to adopt all of this. It might take time, but then we are going to adopt each and every one of these decisions, whether it is genome editing, whether it is GM crop, whether it is policy, or even leading to brown revolution, soil, water, all of these things, we are really going to adopt this. So the time is really going to be exciting. It is already exciting. It is going to be more exciting because not only ICAR, but all other institutes outside agriculture also are really driving in that particular direction. And today your lecture really showed in that way a good roadmap of how we should be leading, how Indian agriculture, how science in agriculture should be leading. So sir, thank you very much once again for delivering such a beautiful and illuminating lecture. And it is always a pleasure, as I said, to listen to Professor Datta. Not only to learn more about science, but also to learn the philosophy in the science. Also visiting those rural areas of those farmers, pallis and whatever he said, and also learning a little bit more about Rabindranath Tagore. So these are additional points, additional advantage of listening to Professor Datta. Only the thing is that we now really have to work. And when I see all the young students, you can consider me also as young, so I am not also so old. So this is now going to be your time. When I talk of these next 25 years, these are basically for your years. These are basically going to be the years of all the students who are sitting over here. And you should always get your inspiration from the presentations of Dr. Datta and of course many more. You are here in this great institute, you will be getting ample opportunities of listening to these great persons. Do not miss these opportunities. All these opportunities will tell you what to do, how to do, what not to do, and how best to establish yourself. And as I said while I was introducing Professor Datta, that your aim should be to have a biodata like of Dr. Datta. And if you can make that as a goal of your life, I am sure not only you are going to contribute for yourself, but also you are going to contribute for this institute and also for Indian agriculture. This institute has served in many, many ways. I do not find that a single policy of Indian agriculture which might not have discussed in this hall, which has got nothing to do with IRA. IRA has been part and parcel of almost anything what Indian agriculture has done. And the remaining of the next 25 years are also going to be the year of Indian agriculture, again led by IARI. And the council looks forward for your contribution. Council looks forward for the achievements of IARI, and council looks forward for whatever is coming out of this great institute. So wish you all the best. Thank you very much for inviting me, and of course special thanks to Professor Datta. And with this, with your claps, let me request all of you to please give a standing ovation for this brilliant lecture, brilliant presentation by Professor Datta. Thank you very much, sir, and thanks to each and every one of you. Thank you.
Thank you very much, sir, for those valuable insights. Thank you again for being with us, and we look forward to many such fortunes in the future as well. So moving ahead, and now a difficult task for all of us, as we have to say thanks to the most conversant speaker of the day, Dr. Professor Datta sir. No present or a gift or any felicitation could ever be enough to pay gratitude in kind. Yet, as a token of love and affection from IRI, we dare to present you a memento, and I request Dr. Himanshu Pathak sir, the chairman of the session, to do the honors, please. Thank you very much, sir. I now request Dr. A.K. Singh sir to present a memento to Dr. Himanshu Pathak sir as a mark of pleasant remembrance of the event and a gratitude from all of us present here. Thank you so much. And with that, I request Dr. S. Gopalakrishnan sir, Head Division of Genetics and also President, Genetics Club IRI, to kindly express the word of thanks for today's event. So,
Honorable Chairman of today's event, Dr. Himanshu Pathak, Chief Guest of today's lecture and today's event commemorating Dr. B.P. Pal's memory, Professor S.K. Datta, distinguished dignitaries of the dais, Professor R.V. Singh, Professor P.S. Birtal, Professor Sadamate, Professor K.C. Bansal, and our former deans, Dr. R.K. Jain and Resmi Akwa, and project directors and heads and professors, ladies and gentlemen, and my dear students. It's my proud privilege to present the vote of thanks at this solemn occasion of celebration in memory of Dr. B.P. Pal. I, on behalf of the Genetics Club, the Graduate School, IARI, New Delhi, and the entire faculty and students of our institute, and on my own behalf, extend a very hearty thanks to Professor S.K. Datta, a mentor of several modern day mentors, and also a pioneer in the research on transformation in indica rice. Now, many of us talk about genome editing, but the basic lies in ability to transform the plants itself, and Professor Datta is known world over for his pioneering research in indica rice transformation, which he did for the first time. Thank you very much, sir, for kindly accepting our invitation and delivering the memorial lecture. And also for you have painted the global canvas of biotechnology research, and also I think through your lecture you have sprayed some colors into the young minds, young students who are assembled here to shine in the future. I would like to extend our sincere thanks to Dr. Himanshu Pathak for gracing the occasion and also chairing, and also a pioneer in another transformation, a transformation in the views of the global eyes on methane emission, and also sharing your thoughts and wisdom leading into Amrit Kaal. I would like to thank our Director and Vice Chancellor, Dr. A.K. Singh, all joint directors, our Dean, our Associate Deans, MOHR, and members of Genetics Club, and also students and staff from all the divisions for helping with the organization of the event. I would like to end with a quote from Dr. B.P. Pal. "The gate of life swings to and fro, and soon, too soon, it closes, and that is why beside my door I grow red roses." This is his quote. I am quote. His life teaches us dignity, courtesy, compassion, and love of beauty. Let us strive to emulate them in all ways of life. Thank you very much to all of you again.
Thank you very much, sir. And with that, we may be allowed for signing off. Thank you all for your gracious presence. Have a great weekend and namaskar.